Hello, fellow foodies, and welcome back. This is Dr. Cassandra Quave, and you're listening to Foodie Pharmacology, the science podcast for the food curious. Today on the show, we're going to address a couple of big questions when it comes to understanding biomarkers of our foods and what that means for our health. And also, we're going to speak with two experts in the field of lipidomics. Now, lipidomics is a very technical term, but don't worry, hang tight. We're going to dive a bit deeper into what that means and how you actually assess lipidomics in different food items. But first, let me introduce you to our two guests. First, we have Dr. Andres Jaramillo-Botero, who is a distinguished professor and scientist specializing in nanoscale science. He's focused on addressing major global challenges such as sustainable ecosystems, climate change, clean energy, health, food security, and symbiotic materials. He does this through integrating engineering, physical chemistry, biology, and computational sciences using a bottoms up approach. Andres has a bachelor's of science in electrical engineering, a master's of science in computer science, and a doctorate in engineering. Has also completed a number of trainings, including work as an invited researcher at Caltech and at NASA's Jet Propulsion Laboratory. He is the founding director of the iOMICAS Research Institute, which is a Rockefeller Foundation periodic table of food initiatives center of excellence that is focused on elucidating and democratizing access to food composition at the molecular level. Now, I'll go into our next guest. Our other guest is going to be Dr. Natalia Vázquez Manjarrez. He did her undergraduate research in human nutrition and food sciences and has a Master of Science degree with honors from the National Autonomous University of Mexico. She also completed her PhD and postdoctoral studies at the University of Copenhagen, where she specialized in nutritional metabolomics. Natalia is a member of the National System of Researchers in Mexico and leads the Metabolomics Unit within the Department of Nutrition Physiology. So, thank you both so much for coming on the show. I know you are both extremely busy with lots of great science happening in your respective institutions. And I'm just really looking forward to learning more about your research on today's show. Thank you very much for having us today. Oh, why don't we start, Natalia, if you don't mind? I know maybe you could start by giving us some basics on the field of, first of all, I know that you work in metabolomics. What is metabolomics before we dive into lipidomics? These are some big concept terms. And then we'll take a closer look at how this ties into your food research.
Yes, of course. Metabolomics is an analytical technique where we use very advanced technologies such as mass spectrometry, and then we applied it into the study of different matrices. This could be food matrices or biological matrices such as urine or blood or feces, swabs from the mouth, etc. And it allows us to be able to detect different compounds that are contained in those different samples. From there, we may get information about specific compounds that have a certain weight, molecular weight. For example, we could detect glycine, amino acids, different types of amino acids, different types of different carboxylic acids that might be interesting to look on, as well as lipids. So lipids would be an area of research that does the same, applying um, the mass spectrometry, but into the study of different lipids and different species within this lipids.
Great. Now, Andres, can you maybe also share with us how are such techniques used in these types of food studies? Like why is it important that we can look at lipids or carboxylic acids? Like what's the importance behind that?
So the primary um, goal of these techniques is really to understand the building blocks of matter. Matter being a food, matter being a living organism, can be an animal, human, any living organism. And those building blocks, what we do is we separate those building blocks in specialized instruments, as Natalia was pointing out. Now, we analyze metabolites as indicative of those small, relatively low molecular weight molecules or compounds that are part of these building blocks. Now, for us to understand this correctly, you need to be able to take a snapshot of that organism. And again, to separate these different compounds and study them in the way they interact with our bodies. For example, if we're consuming food, we're interested in how our compound interacts with your heart tissue, or how it interacts with your lung tissue, and so forth. So it's imperative for us to have techniques that allow us to separate these compounds by different classes. So metabolites are just the low molecular weight, typically below 1,500 Daltons or so. Then we can have proteins, which are larger markers or larger structural elements that are produced by the body. We can have lipids, and we can start, classifying these into the individual functional forms and individual structural forms. So I think that's the main point, is to be able to not only separate them, but understand their biofunction or their function in general. We can use these as spider compounds in industry.
That's great. So, Natalia, when we think about this saying of being, you are what you eat, does how does that apply? I mean, if we're thinking about the molecular composition of a food, do we find that exact molecular composition in our body, or does it go through some sort of transformation?
Definitely goes first into a biotransformation. So we have this adenine, which we would call absorption, digestion, metabolism, and excretion of these different metabolites and compounds that are within a food. A very important thing here is that we also need to talk about our gut microbiota. Then, depending on the different profiling that each person has, then we would be able to create different metabolites. This is something that has been greatly studied, and a very good example of this would be the formation of oral lectins that come from allergic tannins. For example, pomegranate. Pomegranate is very rich in these type of compounds. But when they go into our body and they enter in contact with our gut microbiota, then there are people that are able to create or produce these different types of oral lectins. So these are called metabolites. And depending on these type of metabolites, then it would influence the effect that a person would have on different health outcomes. Mostly, the most studied have been around cardiovascular disease, lipids in the body, and so on. So it is always a metabolite that goes through our system, and then it is transformed as it moves along and then reaches our circulation.
That's great. So Andres, when we think about a lot of our basic science investigations, we're typically taking one variable, right? That's the best, easiest experiment. You have one variable, one item that you're kind of tracking through some experimental process. But as I understand it, metabolomics and lipidomics doesn't just tackle the issue of following one metabolite or one lipid or one protein, but rather a whole series of many different elements. What can you share with us about the complexity of tracking this as it's transformed, as Natalia said, from the food, molecules to what actually it becomes once it enters your body?
That's a great question, Cassandra. So the essential piece that we're after is really structure and composition, molecular structure and composition. If we have those two, and that is how atoms are interconnected between each other, and what atoms are participating in that particular structure to form a three-dimensional compound, then we pretty much know everything about the compound. In the process of characterizing a food or any sort of matrix from a living organism, we're interested in finding all those individual, pieces that make up the puzzle of, for example, an apple. Now, that can be, thousands of compounds. Now, the problem is, since you have different types of compounds, like a protein can be a relatively large molecule. A metabolite typically is a smaller molecule. And proteins, in fact, are metabolites that are polymerized in larger proteins. But the point is, once you break them down into pieces, you can find out of the output of one of these instruments that thousands of compounds are coming out via a mass spectra. which is just a sort of a graph that gives you the individual fragments that make that compound. And we need to be able to identify it and annotate its function. Now, that function can be published already in the scientific literature. Somebody, could have done linear, cell line analysis of how that compound interacts with your body. But it's not always the case. And especially with foods, what we're finding out through the PTFI is that there are a lot of unknowns. I mean, thousands of unknowns. So imagine you're consuming, habitual, regular diet and not knowing anything beyond 10% of what you're eating. So that's scary because we should know more details about what we're eating because, as you were saying, we are what we eat. Food is medicine, and food can actually have a big impact towards the positive or the negative of your health. So our challenge is to deconvolute these very complex graphs into the individual constituents and to then understand what each of those constituents actually does in your body. So if you take the FDA labels that everybody reads on their, packed foods, you'll find a very limited subset of what really makes up a food. And we're not interested in the whole class of saccharides or, sugar, nor in the whole class of lipids or fatty acids and so forth. We're interested in the individual molecules within those classes, how they interact with your body. So that's essential. And it's it's a needle in a haystack type of problem.
Yeah. Well, I can see this goes much deeper than I think when we think about certain foods. So this one has X amount of carbs or Y amount of protein or fats or whatnot. This is really about all of the details behind that messaging and especially around the small molecules. Now, you brought up the example of an apple. And I think that's a fruit many of us are familiar with. Although we know that there are different varieties of apple, like a Granny Smith apple is going to taste different than a Red Delicious. In these sorts of analyses, would you be able to pick up on differences in chemistry then because different apples taste different? And how does that all come together in Italian? Maybe you can elaborate a bit more on like how chemistry affects our what we interpret as flavor.
Yeah, well, of course, there are many other different things and different metabolites that provide these organoleptic properties with different varieties. In this case, you were assessing apples, for example. But we also have to take into account that it's not important only the variety of the apple that you're eating, but also where has the apple been grown. It's not the same a Granny Smith that comes from maybe Washington as that comes from another part of the states. Or in our case, in Mexico, the guava that comes from Michoacan from another one that comes from another part of the state. So it's something kind of like the terroir that you have in the wines that definitely is the property of the soils, the properties of the soil and the characteristics that have been used to grow these different things or the different foods that are going to change their chemical composition and with it, the different tastes. Lipids, for example, is a beautiful example on how many or how these foods may vary and then change their colors, their taste as well. But it is always dependent in something on how is that they've been grown. Of course, chemicals that might have been put into this different food that impact as well the color and different compounds that give these organoleptic properties to the food. And then, of course, that is not really reflected on what a regular consumer would see in the food labels that we see every day. However, I believe that in general, in the food composition, in the nutritional area, regarding the omics technologies, we're going into a new era, which means that not all the time we're going to be able to use the 1,631 lipids, for example, that we're able to detect in our platform now using the PTFI technology. We cannot always provide information that's going to be useful for that. So this information has always to be handled with care. But this is something that we definitely need to start to think about when we are building dietary recommendations and trying to see foods in a different way as a dynamic entity itself that may have an impact in health as well.
That's great. Well, when I think about markers of health, I mean, I'm familiar with the use of the term biomarkers used to, for example, track signals of health or disease in different types of conditions. And that's usually when we think about biomarkers, we're usually thinking, at least in my field, we're thinking about, signals in, the chemistry of the blood or of the urine. Now, I know that you're applying this concept of biomarkers of intake. So the biomarkers, is that really about biomarkers of what we consume and then how it's transformed? Or can you elaborate a bit more on like how do biomarkers play into these nutritional studies?
Yes, of course. So let me first start talking a little bit on the way on how we determine diet health associations.
Okay.
How we usually go around this in a very simplistic way of explaining it is that we would produce this large observational study where we obtain information of the core diet, where we use food frequency questionnaires or we use 24-hour recalls or sometimes use weighted dietary records. However, of those three, food frequency questionnaires are the ones that are mostly applied. And then we try to link it to a specific health outcome, let's say diabetes, a specific type of cancer, a cardiovascular disease, and so on. So after we have that, then we're able to establish which are the risk factors for presenting those diseases or which might be a beneficial factor to avoid the presentation of those. And then from there, we're able to create dietary recommendations that go into dietary guidelines for different countries or create policies that are able or put in place in order to avoid, these type of diseases in the population. However, these different dietary recalls and all these dietary assessment methods that we use have an inherent bias because it is based in the retrospective nature. So you are not always aware of what you eat all the time in every day for, let's say, a month. That is very difficult to remember. And also to assess the portion size that a person consumes. And in my personal opinion, food has as well a moral attitude towards it. So not always a subject that is recruited into an observational study or that forms part of an interventional study will say all the time and be clear the reality of their intake. Now, this is where biomarkers of intake come into play. Biomarkers of intake are compounds or are compounds that are within the food that may serve as proxies of exposure. So what we do is that we go into the food, we study the food, we apply these beautiful and very high-tech technologies such as metabolomics and lipidomics to try and see which are those specific compounds for that specific food in question or a food group, and then use that information to say, this person has consumed the food or not. And then use the information of that specific compound to then do the association with different health association studies. Now, this is crucial for these type of human studies and health studies, because then by using them, we will be able to create more robust associations and be more clear on what is it that the dietary factors can do into impact these different health like diabetes or other diseases that I have mentioned. There is a
Wow. So
very oh, sorry, yeah, go on. Yeah.
no, so I'm just thinking like how this could be applied. Let's say if you had blood samples from 1,000 patients that are diabetic, you could then measure and say how much broccoli have these people actually eaten. Is that kind of or cruciferous vegetables? You could look for those signals in their samples. That's fascinating.
Yeah. I mean, it's definitely fascinating. You could go about the question in a different way. Let's say that, for example, in my country, as you know, we have a very high level of intake of soda, right? Or other foods, ultra processed foods. So you can go about into your research question like this, like, what is the impact of the intake of soda in the Mexican population on diabetes, for example, in type 2 diabetes, or on children obesity, for example. So what we do is that we go at it the other way. So we try to find these biomarkers that would tell us the exposure to that specific food, and then be able to determine the risk of that exposure and the appearance of these different diseases.
Interesting. Great. Well, Andres, I was hoping maybe you can tell us a little bit more about how lipidomics contribute to this understanding as well. So I'm assuming when we think about sodas, that's more of a metabolomics question, right? You're able to look for small molecules. How does lipidomics help us to better understand diet and perhaps even inform dietary interventions?
So, Cassandra, the first thing is in our lab, we don't do lipidomics at this point. We have not onboarded lipidomics from the PTFI effort. I think uh, Natalia is the only one focused right now on lipidomics.
Okay.
We're
Very
doing, we're,
mixed up.
we're doing metabolomics, we're doing ionomics, we're doing fatty acid analysis, which is a subset of lipidomics, and we're doing proteomics as well. So I think out of the discussion that you two were having just now, I can add the following that I think is important to state. One is each individual is a world apart from others, and what distinguishes them is the way they interpret how these molecules interact in our health, both to taste and flavor and so forth. We have taste receptors. Those receptors we have in different quantities from person to person. So how we interpret food changes from a person to another one. The other thing that chemically transforms food significantly is the way we prepare food. And, that changes from Mexico to the US to Colombia to any other place in the world. The way we thermally modify food gives us another spectra of high-end chemistry. That brings a different level of complexity into the picture. Right now, we're focused on understanding raw food. Most of them are coming from, your typical agricultural crops. And there's also, in that case, what you were mentioning, which is the phenotypic variants, how these different, species, they might have the same sequence or genetic sequence, how they express differently under different environmental conditions. What we do to analyze and to classify is we need to go, resort to statistics. We basically look for distributions that allow us to do under these, case studies, what the mean or average would be for that particular population. And then you can do an association study. But again, the individual response may change significantly. I can be, predisposed to express diabetes type 2. Somebody else might not, even if they're having the same diet or super processed food or ultra processed food, they might not express it. And that's when you need to go into the details of the individual's biological composition and how it's interacting with food. And eventually that's going to come down to personalized nutrition in a way that has not been done so far, which is, really we need to hone into the structural components of what we're ingesting and how that is affecting my health. Eventually we'll get there, but for now we're in this paradise of immense information that we need to decode.
That's such a good point. And I mean, as you're speaking about chemistry of foods and differences and terroir, differences and varieties, I'm also drawn to this difference that we can see also in kind of industrial production of our vegetables. Here's an example. So I am a modest gardener. I'm able to grow a few tomatoes in my backyard each year. Uh, but those tomatoes taste completely different than the tomatoes I can buy at the store. I mean, right now I can go to the store and buy tomatoes, which are clearly not, we're recording this in late November. In the state of Georgia, you wouldn't typically have tomatoes growing at this time of year, but I can purchase them at the store. They've probably been grown in a different part of the world, picked green, gas to force them to turn red. And so they look beautiful, like physically, typically they look very similar to the tomatoes I grow in my garden, but their flavor profiles are completely different. So how can we use these omics tools to better understand, or can we use these omics tools to better understand the differences and also in how, not just how foods are cooked, but also how they're grown? Like, will this give us insights into health values? Like, is a tomato a tomato? Is a tomato actually still healthy if it's grown in one way versus another?
Yeah. So uh, I, you would have to go to what the whole PTFI effort, the Periodic Table of Food Initiative is. Uh, the fundamental idea behind this is let's see if we can extract the building blocks of foods worldwide. If we can democratize the compositional information about these foods, and if we can statistically correct those distributions based on the location of where the food was grown using metadata, then we can have information to distinguish if what we're eating is the same tomato, the same quality of tomato, or if you're just having a different taste experience at that point. Because you could individually, you could have different taste experiences with the same tomato. Um, Again, what PTFI is pointing towards is let's build a large international database where at this point we have 9 centers of excellence distributed throughout the different regions of the world where you get phenotypic variances. Um, if we can hone in into how these phenotypic variances occur for the same species. So if I'm eating, uh, Japonic rice here in Colombia, how is that different from the Japonic rice in Japan? Um, we can now start doing that. That we can now start playing this game once we build up enough information into the database. For now, we're at I would say, the cusp of starting a new era in which we could eventually uh, describe diets in a personalized manner from that database. Again, the variables are infinite, and I don't think we will be able to do anything without having a large set of, you know, uh, samples into that database of the same tissue grown in different places. So if we don't have enough statistical sampling, it's hard for us to make conclusions about what might be causing, for example, the existence of high prevalence of diabetes type 2 in Mexico or cardiovascular disease. And if you add other variables that are coming into play now for the design of foods, you might be interested in looking at, for example, how a particular cultivar is affecting soil and is affecting the atmospheric conditions of your region. Are you regenerating soil with the type of crop you're putting into it? So there's added complexity, and that's going to come by the way of looking at other issues that are related to agriculture, including the sustainability of food production when you're deforestating for putting more animals that grow meat or milk, when you're putting more plants that are not necessarily very good CO2 sequestering, they don't have good sequestering capabilities for CO2. So you need to have other variables now come into play when deciding what to eat. And I think that's adding to a lot of complexity. Uh, a good thing now is that we have big data analytics capabilities. We focus a lot on developing those capabilities in a way that will allow us eventually to get to the understanding of individual foods and what the differences might be when one individual consumes it or another, or where it's grown.
Right. Well, I'd love to hear more about how each of you are working with the periodic table of food initiative, the PTFI, and kind of how this is, what directions it's taking your research. Maybe Natalia, I Andres mentioned you're doing a lot of lipidomics now. Like, how is all this contributing to the overall mission of PTFI and democratization of this data?
Yeah, well, our how we work with the PTFI is they've been democratizing the different methodologies for different omics. So they have developed lipidomics platforms, which we are now onboarding in our metabolomics unit, as well as on-target analysis, proteomics analysis. They're also doing ionomics and things, which is what Andres has onboarded in his lab. But there are many other labs from Andres that were 9 different ones distributed in different continents. And each of us had the responsibility of developing different demonstration projects. In our case, in our Center of Excellence, we are going, our demonstration project is BIAME, which stands for Biomarkers of Intake of Mexican Food, as well as MAPAME, which is the mapping the metabolomic composition of different Mexican foods. So under that umbrella, we are trying to find those biomarkers that we were mentioning before. For those foods that are highly consumed, we're starting with six, which would be nopal, guava, mango, corn tortilla, amaranth, which is a beautiful project that we are also partnering with our UC Davis collaborators in order to try and understand the proteomics and peptidomics behind it, and then try to figure out what are those specific compounds that may constitute biomarkers of intake. I think the PTFI as well has been doing a beautiful job in democratizing these different tools that otherwise it would take in a lab such as mine years to be able to onboard. So we very much act like a very nice network of different platforms where I'm able to send food samples to, for example, CSU, where they're currently being analyzed for their own targeted metabolomic analysis. And maybe Andres, if he needs a specific lipidomics analysis on a food that he's growing, he may send that to my lab where we can collaborate and then do all these different results and then upload that into the PTFI database with the completed metadata. So we're trying to look into food in a different perspective. We're putting food in the center of different problems around the food system. So how is it that this particular food impacts planetary health, as well as how it impacts human health and everything in between, so that it makes it, in my opinion, a unique network of science, of scientists that are trying to solve these different questions.
I really like that approach of thinking about health, not just from the perspective of human health, but also planetary health, because I think you really can't have one with the other. That's so true. Andres, I know that your role is also in leading this one of these centers of excellence. What can you share with us about the actions taken at the center of excellence and how you network with other labs?
We're lucky enough to be part of a very large network, as you were mentioning, not only of these centers of excellence with PTFI, but we've been involved for many years now with a large set of agricultural research centers throughout the world or in different places of the world, including one that is very important within the PTFI, which is CIAT. It's an international center for tropical agricultural research. And CIAT is part of the subsecretariat with the American Heart Association of the PTFI program. So we've been involved in doing different demo projects within our lab, and that one of them that I can tell you is very interesting is confronting cultural heritage behind food with scientific evidence. For example, we have a large set of exotic fruits in Colombia. More than right now, edible fruits in Colombia, you can find, I think the number is around 3,800. So that's quite a large set. In spite of that, we're only consuming a fraction, less than 24% of that. Why is that? Of the exotic fruits that are not expressed anywhere else, we've been doing analysis of those that are very, very attached to the cultural, heritage of populations, communities, small communities throughout the Andean region. And I can tell you, I can give you a very interesting anecdote. One of these is a palm tree fruit called chontaduro. We call it chontaduro in Colombia. It's very popular. It was brought by the Black communities to the Pacific region of Colombia, and it's very popular among those communities and among the cities that are alongside the Pacific region of Colombia. It's sold with extraordinary aphrodisiac properties, and they had been exaggerated for years. And we decided, okay, let's hone in. Let's try to see if we can find metabolites that are associated with, aphrodisiac activity, indirectly or directly. And what we found is that indeed it has several indirectly associated metabolites with this biofunction, but they're not in the piece of the fruit that we eat. They're not in the pulp. They're in the seed. Nobody consumes the seed. If we wanted to consume the seed, then we need to look at the composition of the seed, and we did. And we found, for example, that the omega-6 to omega-3 relationship in the seed is too high to consume. You would have to maintain that ratio to about 4. Omega-6 is a pro-inflammatory fatty acid. The omega-3 is anti-inflammatory. This seed can have up to 180 to 100 in that ratio. So we now are starting to get to the point where we can tell history, okay, what you've been consuming might be true, or it might not, and it might be affecting your health. And that's where we're getting now. Now, the complexity of how do we inform these findings is also interesting. We had to hire an anthropologist to describe that in a way that would not affect the communities or affect the commercial activity behind those schools. So we've been for years dealing with some of these problems, and we had a large alliance called the ÓMICAS Alliance. We were 17 institutions, including Caltech, for which I work with for many years now, and it's been very rich to find how different disciplines understand biology these days, and biology of food and biology of fuels. So these are alliances that have, pretty much every field under the sun in terms of disciplinary activities. And we've been fortunate enough to have, rich alliances of more than 300, 400 people, each one considering these aspects from their own disciplinary point of view. We're also working with FIAT on characterizing the worldwide gene banks that they have for pulses, basically bees. They have more than 38,000 accessions of different bees that are consumed worldwide. What we're finding is that if we look at the proximals, what characterizes the bees, they're almost identical in total protein content, total carbohydrates, total lipids, and so forth. But as soon as we go deeper into the metabolites composition or into the individual fatty acids, ions, and so forth, they start separating clearly into very different species that are now explaining us why a particular D is heavy on your stomach, why, lectins might be affecting how you consume beans. So these are very interesting findings that I think if it were not for the network that we built, and that is very active in fundamental science all the way to translational science, we would not be at the point there at which we are now. So everything is converging, but it's going to take a few years until we get that full periodic table map of metabolites, protein, and whatnot. We're now in an effort with CIET and others, including, these are efforts funded by the Bezos Earth Foundation and by the Bill and Melinda Gates Foundation to, for example, look at the environmental impact of having livestock and their methane producing activities during enteric digestion. The whole deal comes from, we need to understand the forages at the molecular space to understand what are the compounds that are actually activating this methanogenic pathways within the cow's stomach, including what the microbiome in the livestock is doing. So if we can actually pinpoint these anti-methanogenic compounds, then we can edit the forages to produce forages that have no CH4, no methane emissions. And that's been tested. We can, I think it was UC Davis that they experimented with algae, giving the animal algae, and they brought down the methane production by about 80%. So I think we're now at the point of, again, coupling a lot of problems that are coming from our food system that it is now basically going beyond the boundary of just food science. And we're getting into environmental science, and we're getting to other aspects that are important for the food system.
No, I love that. I think that there's so much that we can learn from having a better understanding of, it sounds so simple, just the building blocks of our food, though it's very technically challenging to get at that, I'm hearing. Um, we're almost out of time, but before we wrap, I would love to hear from both of you about, where do you see the future of food going? I mean, equipped with this knowledge, do you see this being a tool towards a path to better sustainable ag, or discovery of new superfoods, or something else that I can't even think of right now? I'm just curious, like, where do you see the future of food going once we're fully equipped with this type of knowledge? Maybe Natalia, you can start.
Yeah, well, I definitely see the future of food coming to the discovery of new superfoods. But I also see a way of which different countries and populations can introduce biodiversity into their foods, taking into account the foods that originate in their countries. And with that, be able to construct dietary patterns that are actually useful for that particular population. Let me give you an example. We know that the Mediterranean diet has repeatedly proven and very well established to ameliorate different diseases, in this case, cardiovascular disease, so on. However, in my practice as a dietitian, when I see very, very few people are able or to access for olive oil or for different foods that are specific for these dietary patterns. So in my mind, where we can take these vDomics technology and then apply it further, what if I had the information of this plethora of foods that originate in my country? For example, chia seed, almond seed, different forms of preparation of different vegetables, foods, even meat, and different fishes that we can actually, or that are accessible in my country. And through a data-driven approach, construct the data, build up a diet that may resemble the composition of that have already been proven to be beneficial for a specific disease. So with that, I may be able to create a dietary pattern that is actually useful for Mexicans in our case, or for different countries in Africa in their case, and so on. So I believe this is a multitude of places where we can take food and food research in the future to the construction of more precision nutrition, precise dietary treatments for different people as well as for populations, and use this information to do that in a more sustainable way, in such that we are able to have a better planetary health, bring more sustainable sources for proteins, for example, as Andres was mentioning, which is obviously a very problematic area at the moment, as well as to try and rescue other foods that have been, eclipsed by the intake of others, such as amaranth that have been eclipsed by wheat or by maize in our case.
Yeah, I mean, I like that because you're really factoring in this concept of food sovereignty, which is really about, food security plus cultural relevance of foods, which I think that's really cool. I hadn't thought about that as an aspect of this, but I clearly see it, like the value of that. Andres, how about you? What are your thoughts? Future of food with this knowledge?
Yeah, so I think it's a great question, Cassandra, and I'm very, just heuristic in the way I think about research and beyond being very optimistic. So in an analogy to the periodic table of elements, we have 118 or so elements in the periodic table. Out of those, 55 are natural, but only 22, typically 22 form organisms, living organisms. And if we think of possible combinations, permutations of those elements to produce what we have on Earth and in the universe in general, I am very optimistic in that if we find a sufficiently complete set of compounds you know, constituent elements of food, we should be able to design foods, you know, build molecular labels out of the individual compounds that we can find from different sources, and eventually start to relate that to the genotypic nuances of an individual and how an individual can actually absorb and optimize the absorption of food for, to maximize health. That is, I think, the future of these efforts, albeit they're not short term. I think we should be thinking about these in the relatively near term. Now, I am not convinced that we will find superfood, meaning one food for all. I don't think that's the way it works. And that's the beauty of it, actually. Otherwise, we wouldn't take food as a social activity as well. I think food has many, many edges to it, not only the nutritional aspect, but the cultural aspect. We need to be able to enjoy eating food. So I don't see food being packed in a pill and then, as astronauts will take them. That's not the future that I see, but I do see that we can supplement our food based on our individuality, our individual genotypic expression. That's the future I'd like to see in the near term. And I'd also like to see that, food needs to become a commodity that is accessible to everyone. There's a very clear distinction in what we call today the global South and the global North. There's a poor, non-geographically meaning South of the world, and a richer, more developed North. The question is, how do we get food to everyone without distinction of socioeconomic status? I think that's also a point that we can address with the PTFI effort, assuming that we can, start producing these foods that are well equipped to grow anywhere in terms of crops. And we are now at a point in which we can start stating, okay, this particular species, you can do selection of species and say, okay, this will grow very well in South Africa. This will grow very well in South America and other places that have little access to biodiversity. Fortunate for us in the tropics, we're very biodiverse. Probably the north, the actual geographical north and south have a harder time producing biodiverse foods and individuals. So I think eventually access has to be a key point of these things. What PTFI is promoting is the idea that we at least have access to the compositional data. And I think that's the first big step to democratizing access.
Amazing. Well, I think there's much to be said for access and the ability to promote and advance science, and a lot to be said about the value that this might have for personalized food care, health care, medicine. I don't know. We talk a lot about personalized medicine, but I think our health really starts with our plate. So I think this could really help us make some better choices in the future. I want to thank you both so much for coming on the show. What you're doing in the lab is fascinating, and I'm excited to see the studies that come out of your groups and are shared through the PTFI platform and network. And thanks again for coming on the show.
Thank you, Cassandra.
Great.
Thank you very much for having us.
Thanks. You've been listening to Foodie Pharmacology, the science podcast for the food curious, recorded for you today on Squadcast. I want to thank our listeners for tuning in each and every week. You can find this and all of our other episodes on the Teach Ethnobotany YouTube channel, or you can head over to our podcast website at foodiepharmacology.com, where we have links to a lot of fun swag and our past episodes. Lots of great details to be found there. I want to thank our producers, to Rob Cohen and Christine Roth, for putting on a great show. And thanks so much for listening. Stay healthy out there, and we'll see you next time. This Foodie Pharmacology podcast is part of a special series co-designed with the Periodic Table of Food Initiative, also known as the PTFI. The views and opinions in this podcast are those of the presenters and represent the synthesis of science. The PTFI is a program of RF Catalytic Capital managed by a collaborative team at the American Heart Association
and the Alliance of Bioversity, CIAT, that seeks to advance our fundamental understanding of food composition by providing tools, data, and training to scientists across the globe so they can better characterize the quality of the world's edible biodiversity. The PTFI's ultimate goal is to advance data driven solutions in the food sector for the health of people and the planet. Funding for the PTFI is provided by the Rockefeller Foundation, the Foundation for Food and Agricultural Research, C-Rave Foundation, Fourfold Foundation, and Atria Health Collaborative.